Thank you. I should say this is a new topic for me. I've done a bunch of thinking about some technical aspects of this, but I haven't given this talk before, so I hope it will be of interest, and apologies if it's quite technical in places. I'm going to start by talking about how many knockouts we're likely to be able to discover in the next few years, given planned projects in this country. Secondly, how we might accelerate the discovery of new knockouts and their characterization, and then about how to validate predicted loss and the problems in actually predicting which loss of function variants will truly lead to loss of function, and how we might try to tackle these. So, as has already been mentioned, loss of function variants are really enriched for low allele frequencies, and this is particularly the case because most of them have probably been under negative selection. In this landmark paper of Daniel MacArthur's in 2012, this became clear, but they only had 200 individuals, and so necessarily they were overestimating rare allele frequencies. We now know with much larger data sets that actually the vast majority of LOFs are present in less than 1 in 1,000, or actually less than 1 in 10,000 people. We can calculate the expected frequency of knockouts, that is, individuals who are homozygous or compound heterozygous for a loss of function variant, using two parameters. One is the cumulative frequency of LOFs in that gene, so what fraction of haplotypes in the population carry a loss of function variant. And the second parameter is the fraction of individuals who are autozygous at that gene, meaning homozygous due to recent consanguinity. So in this equation, the first bit is the knockouts that are due to people who are that are in the autozygous fraction, so these will all be homozygous by definition. And the second is the chance of a knockout when you're not autozygous, and those will be mostly compound heterozygous. So in a non-consanguineous population, the vast majority of genes have got an expected frequency of knockouts between 1 in a million and 1 in a billion people. That's the purple distribution over here. So obviously, these are very rare. The median gene has an expected knockout frequency of about 6 per billion individuals. In contrast, if we were to go to a population where everyone was the offspring of first or second cousins, then the median gene has an expected knockout frequency of about 5 per million individuals, so much, much higher. And these are, of course, mostly homozygous. In this plot, I'm showing the number of genes we'd expect to find with at least one knockout, given the sample size on the x-axis, in two different populations. So what you can see is that with 500,000 individuals from UK Biobank, which is mostly non-consanguineous, we'd expect to find about 2,000 genes with at least one knockout, whereas in a project, a cohort of, say, 100,000 people in which everyone was the offspring of a first or second cousin, we'd expect to find more like 7,000 genes with at least one knockout. As I said, in an outbred population, most of the knockouts will be compound heterozygous. And so in order to demonstrate that they're knockouts, one would have to phase them, which could be done potentially with long-read sequencing. Of course, it can be done with trios, but those often won't be available. And it could potentially be done by kind of follow-up RNA sequencing, although that has some difficulties that I'll talk about at the end. Now, for many genes, it's likely we're going to need several knockout individuals for the same gene in order to attribute a phenotype to that gene, based on association testing alone. And so here I'm showing the expected number of genes we should expect, the expected number of genes with at least 3 or at least 5 knockouts given the sample size. So you can see these curves are shifted to the right, of course. If the number of knockouts that we found is very tiny and they have some unusual biology or unusual phenotype, it's likely they're going to require substantial functional validation to actually prove causality. There's been a lot of enthusiasm, and this was mentioned earlier, about founder populations for discovering knockouts, and the Finnish population is particularly well studied. As a result of the bottleneck, some variants that are rare in the original population can drift up to a higher frequency. So this plot from this paper a few years ago is just demonstrating that there's an enrichment of loss of function variants with a frequency of between 0.5% and 5% in the Finnish population. So that means if you happen to be interested in one of those knockouts, you'd have a higher power to detect associations, which is a good thing. On the other hand, these bottlenecked populations also have fewer variants because the variants have to either have been present in the original founders, or they have to be very recent and have occurred since the bottleneck. This is illustrated in these two plots, where you can see on the bottom there are about 11,000 genes that had 0 loss of function variants in about 10,000 Finnish people from Nomad. In contrast, if you take about 10,000, or the same number of non-Finnish Europeans in Nomad, there are only about 7,000 genes with no loss. So there's this kind of trade-off with bottleneck populations. There are more variants that have a higher frequency, but there are fewer variants overall. So some work that my group's been doing recently to characterize the population structure of British Pakistanis has revealed that actually this population has multiple subgroups, which we already knew, but what we've shown with the genetic data is that these are, many of them are very bottlenecked, even more so than the Finnish population. In this left-hand plot, I'm showing the IBD score, which is basically the average length of IBD segments shared between two individuals from the same population. And these are all tribal, ethnic, or kind of clan groups with self-declared from the Born in Bradford project. And the score, it has been normalized to the Finns. So the Finns have a score of 1, and you can see all these Pakistani groups have a much higher IBD score, implying that they're much more bottlenecked. So this suggests that there are probably going to be lots of founder effects with loss of function variants having risen to a higher frequency. Then additionally, because of the high rates of cousin marriage, those variants will be even more likely to become homozygous, of course. We are still studying the extent to which these bottlenecks are independent versus happened to some extent in a common ancestral population, which will determine the extent to which the data set of the whole kind of has fewer variants than other populations. There are kind of technical reasons why it's difficult to assess this at the moment. But it's likely that these bottlenecks must be to some extent independent, because otherwise they would all, the groups would all look the same. So let's just take a step back a bit and think, supposing we could sequence 100,000 East London genes in health individuals, of whom about 20% were offspring of first or second cousins. Then there's about a 37% chance of finding more than one homozygote for the same variant that has a frequency of 1 in 1,000 people. So that's not terribly high. Of course, we might well detect different homozygous variants in the same gene, which would be useful. But supposing we did want to find multiple individuals who carried the same biallelic loss, well, the most sensible thing we could do would be to recruit their siblings, who of course are IBD2 for 20% of the genome. And this was nicely illustrated in the PROMISE paper that was raised earlier, where one of the individuals who was homozygous for this ApoC3 knockout was actually married to his cousin, who was also homozygous for the same knockout. So all 9 of their children were homozygous for the same knockout, which was extremely useful for their recall study. So David already told you what the results were, but the key point I want to make is that the most efficient design for finding and then characterizing these knockouts would be to sequence large numbers of unrelated individuals from a highly consanguineous population, but to make sure you consent them to be able to contact their relatives and ask them if they want to participate too, if you find an interesting knockout. So, as I think I've already made clear, unless we've got huge sample sizes or family-based cohorts, we should expect that this is going to generate a lot of n = 1 experiments. If the function of a gene's already known or suspected, then it might be possible to kind of jump straight to the confirmatory experiment to work out what the result of that knockout is. But firstly, that's kind of less interesting if you already know the function of the gene anyway. And secondly, I think we should require the same burden of proof in ascribing function to these knockouts that we do for Mendelian disease genes. So if you were to sequence a person with a rare Mendelian disease and found they had a knockout in a particular gene, you definitely wouldn't say that was causal unless you had multiple other individuals or they had many relatives and the variant segregated and so on. And we should treat benign and beneficial knockouts in the same way. If the likely consequence of the knockout isn't known, then a fruitful way to proceed would be to do a PheWAS, a phenome-wide association study, using electronic health records. But we should remember that, of course, this is going to have a substantial multiple testing burden, so we're going to need to see pretty low p-values to be convinced of association. So again, we're going to need to have large sample sizes and multiple individuals with the same knockout. Of course, if the effects are additive, then we're going to have more power than if they're recessive. Something that's been raised several times already is the utility of these knockouts for looking for potential side effects of pharmacological inhibition. But we should remember that it's going to be difficult to rule out later onset side effects, particularly with small sample sizes, given the general age distribution of these population cohorts. Of course, knockouts can be very useful in allelic series experiments where you collect people with multiple different types of variants in the same gene with different effects. And one nice example of this is given by Robert Plenge, and that's the TYK2 gene, which encodes a tyrosine kinase. Knockouts in this gene result in immunodeficiency, so clearly complete knockdown of the gene would be very toxic. But what they showed in this paper is that a subset of missense variants in this gene are associated with reduced risk of rheumatoid arthritis and other autoimmune diseases. And these missense variants have a partial but not complete loss of function effect. They showed that homozygous individuals with these missense variants had the greatest protection from rheumatoid arthritis, but when they did a PheWAS of 30,000 individuals, they didn't find any convincing associations with nasty side effects like infection rates. So this suggests that this might be a useful drug target for these autoimmune diseases. And this is just to emphasize how loss of function variants are likely to be most informative when combined with other variants in the same gene that have differing molecular effects. So to increase our chances of being able to actually work out the consequences of knockouts, it's going to be very important to have electronic health records to allow us to do PheWAS or specific targeted queries. It's also going to be critical for many genes to be able to recall individuals because there'll be phenotypes that don't show up in the health records or that maybe can only be elicited in an experimental setting. These are likely to have multiple logistical challenges. The first one being that, of course, you require individuals to agree to come in, and if you have only one or a tiny number of individuals with very valuable genotypes, I think we have to think about what sort of incentives might be appropriate, if any, to really encourage these individuals to come and participate in our studies. Assays that need fresh blood are always a challenge because you have to get the individual in on a working day at the right time and this kind of thing. As are assays that are particularly plagued by batch effects. I'm thinking, for example, of assays where you want to perturb immune cells with cytokines and see what happens. This kind of thing can be particularly challenging in these studies because, of course, you need to bring back multiple individuals at once with different genotypes on the same day. Another thing to bear in mind is if the phenotype you're looking for hasn't been characterized in a large number of healthy people already in the literature, what's the appropriate number of controls to do to convince yourself that this individual's really an outlier? And is it even practical to do that in a recall study? And finally, but not least, something I haven't yet talked about is the predicted loss of function variant even real? Does it really result in loss of function? We know that these variants are highly enriched for false positives, and there are multiple different error modes. So there are three main ways that a predicted loss of function variant might not be real. One is that the variant's not real. It doesn't exist in any of the individuals in your sample, and it's a sequencing or mapping or variant calling error. The second is the variant's real, but this person's genotype is wrong. For example, you might call spurious homozygous genotypes because the person has low coverage. And the third is that even if the variant and the genotype are real, the annotation might be wrong. And there are multiple ways that it could be wrong. For example, you can have rescue errors where another variant nearby might kind of cancel out the effect of the original variant, like an in-frame, like a frame restoring indel or a splice site rescue nearby. Furthermore, you can have impact errors where maybe the loss of function variant only affects the minority of transcripts or only transcripts that are expressed at a very low level across tissues. So this screenshot I'm showing you is from the LOF curation portal, which has been built by Daniel MacArthur's group at Broad, which allows you to do manual curation and look at the sequencing reads as well as the annotation in the UCSC genome browser to try and pick up these multiple error modes. I should say that a lot of this could probably be automated if somebody got around to writing the code, but some of them I think it would be really quite tricky code, and there probably will always be a place for some manual curation. So they applied this manual QC to all of the homozygous LOFs in Nomad, and they found that actually about a third of them were spurious upon manual curation. I think it's a bit of a wake-up call to all of us that most of the numbers that have been reported today are just based on bioinformatic filtering, and a third of them are probably wrong. One of the main features that's used to predict whether an apparent loss of function mutation is going to result in nonsense-mediated decay is the distance from the end of the transcripts. So about 50% of the variance in the efficiency of nonsense-mediated decay is explained by whether or not the premature truncation codon is in the last exon or within 50 base pairs of the last exon-exon junction. But in this paper, they analyzed various other features of transcripts and found that they could explain another 25% of the variance in nonsense-mediated decay efficiency. But there's still another 25% that's not explained. So basically, we're still not great at predicting bioinformatically which loss of function variants are going to, which predicted loss of function variants are going to lead to nonsense-mediated decay. So I think there's definitely going to be a place for high throughput validation of these, for example, RNA sequencing, which will be useful for genes that are expressed in blood, at least at a reasonably high level. However, there will be a subset of predicted loss of function variants which don't lead to nonsense-mediated decay, but lead to truncated proteins which are maybe unstable and get degraded. So we need high-throughput proteomic methods, ideally, for assessing these. A couple that have gained traction recently have been Olink and SomaLogic, which are based on binding antibodies or aptamers, small molecules, to the proteins. And these have some advantages, but definitely some disadvantages. So they really for proteins in plasma, they won't work if the aptamer or antibody binds downstream of the premature truncation codon. They wouldn't be expected to be informative. So in some ways, mass spectrometry-based methods would be more reliable, but they're also not so sensitive and don't work so well for proteins that are expressed at a very low level. So I think it's likely that a lot of proteins are going to require specific targeted assays to assess whether premature truncation is leading to degradation. Despite the problems with these methods, I think there's still a lot of value in applying them at large scale to, say, all of the knockouts found in Genes and Health or the UK Biobank project, or indeed Genomics England, not just for trying to validate knockouts, but also along with other omics, to allow us to functionally characterize the results of these knockouts. So what are their consequences at a molecular level? And that might give us ideas about specific hypotheses we could test with PheWAS or in specific recall studies. So in conclusion, I've shown you that for more than 50% of genes, we're still going to require sample sizes about 300 times bigger than UK Biobank from non-consanguineous populations in order to find even one knockout. But about 200,000 offspring of first or second cousins would give us equivalent power. Secondly, a single knockout often won't be enough to ascribe function, and so it'll be critical to be able to recruit relatives, which is the most efficient strategy for finding additional knockouts. Thirdly, it'll be very important to be able to recall individuals and access electronic health records, but these will still have some major challenges associated with them. And lastly, determining which predicted loss really lead to loss of function is an essential and non-trivial step to do in a high-throughput manner. RNA sequencing and other omics can be helpful, not just for validation, but also studying the molecular consequences of these variants. Thank you.